Welcome everybody to Lesson 3, where today we will discuss how HLRCC is inherited and why your chances of being born with this mutation and potentially passing on to your own children is a 50-50 chance. Now, HLRCC is known as an autosomal dominant disorder. And when we say autosomal, we're referring to genes located on any of the numbered chromosomes. And as we can see here, this includes 22 pairs of chromosomes highlighted in blue. Or another way of saying it, it's every chromosome except the final pair of sex chromosomes, or those XY chromosomes at the very end. And when we say a genetic disorder is dominant, this means that only a single copy of the mutated gene is necessary for the disorder to be passed on to the child. This single dysfunctional gene can be inherited from either the mother or the father. Importantly, all autosomal dominant disorders have a 50% chance of being inherited. However, inheriting a dominant disorder does not necessarily mean you're destined to suffer all or even some of the effects of the disease. The proportion of the patient population that will exhibit the symptoms of a genetic disorder is what is known as the penetrance of the disease. In the case of HLRCC, there is an incomplete penetrance of the clinical manifestations of the disease, meaning not everyone with the FH mutation will go on to develop health problems. Some patients will go on to express severe symptomology, some patients will have mild symptoms, and others may have none at all. This unpredictability is true even within families that share the same exact mutation. And this variability in symptom expression is called variable expressivity. And based on patient population studies, researchers are able to give estimates of how likely HLRCC patients are to develop specific symptoms. Now, when I look through the literature, in the latest, largest studies, it shows up to 70% of HLRCC patients will go on to develop skin leiomyomas, or those tumors on the outside of our body. In women, 79 to 100% will develop uterine leiomyomas, also known as uterine fibroids, and it's estimated that 15 to 30% will develop an aggressive type of renal cancer. It's also important to mention that the penetrance of these symptoms increases with age, and that's simply because as more time passes, there's just more opportunities for things to go wrong. Now, it should be pointed out that different studies on different patient populations will report different percentages, hence the wide range of variability in these figures. Also, every week, more and more patients are being diagnosed with HLRCC, and what was once considered a very rare disorder is turning out to be more common than originally expected, meaning as time goes on and research expands, these percentages are going to continue to probably change. So taking this all together, this comes back to a common saying where genotype doesn't equal phenotype. And this is just a fancy way of saying that a person's genetic code, or genotype, doesn't perfectly predict their observable characteristics, also known as their phenotype. There are known and unknown variables that can influence a patient's phenotypic expression of disease. These can include environmental factors like pollution, radiation, lifestyle choices. These all come together in determining the outcomes of what happens with people with these types of mutations. So it's this combination of biological complexity with the extremely limited patient population that makes it so important for as many HLRCC patients as possible to join as many research studies as possible, where hopefully we can begin to tease apart these complex interactions and figure out why some of us suffer worse fate than others. Now, to look more closely at why there's a 50% chance of inheriting HLRCC, let's zoom in and look more closely at chromosome 1. As we could see in the previous chromosome picture, each person has 2 copies of chromosome 1. You inherit one from each of your parents. Because humans have a pair of each autosomal chromosome, we are what are known as a diploid organism, where dip means 2. This means that each chromosome 1 will have its own FH gene, here at the Q arm at band 43. These multiple copies of the same gene are what are referred to as alleles. And typically, everyone has 2 alleles for FH, and they should have identical genetic sequences. If these alleles are identical, they are referred to as homozygous. However, in HLRCC, one of our FH alleles is mutated. In this example, the mutation exists on the chromosome inherited from the mother. This mutated allele is called a heterozygous mutant. Heterozygous meaning the genetic code between the 2 alleles isn't identical anymore like it should be. The normal allele containing the correct genetic sequence for FH exists on the chromosome inherited from the father. Since this is how the allele typically appears in nature, it is known as a wild-type allele. So to sum up, in HLRCC, each patient has a good and bad copy of the FH allele. Let's now see how this scenario plays out when someone with HLRCC sexually reproduces. Here then we can see how sex cells, in this case the mother's eggs, are given the genes that they will eventually pass on. And when sex cells are being made, they can only take one of the two alleles with them. So for a quick review, we can take a look at this diagram about how that happens. If we remember, each parent has 46 chromosomes, and the respective sex cells will contain half the amount of chromosomes necessary to make a new child. So when the sperm and egg come together, each of their 23 chromosomes pair together, and we get 46 total for the new offspring. So looking back, we can now see how we split up our alleles. And because there's only two alleles to choose from, 50% of the HLRCC's patient's sex cells will contain a mutated variant, and the other 50% will contain a normal wild type. We can then see how these odds play out when the egg cell meets a sperm cell. Using what's known as a Punnett Square, we can now determine the relative probabilities of each egg and sperm pair. In this example, the female has HLRCC, one mutant allele with a red X, and one wild type allele with a blue check mark, while the father has two wild type alleles, so no disease. This 2x2 sperm egg setup means that there's 4 unique possible combinations that can occur. Let's watch how each of these plays out. Our first potential pairing shows an egg with a mutated FH allele fertilizing with a sperm with a wild type FH allele. The way this is usually written out is with a positive and negative signs, where FH negative means a mutation and FH positive means wild type. So in this case, FH negative / FH positive is a sign for HLRCC. Our second pairing shows 2 wild types together, so 2 FH positives. Our third possible combination is another mutant wild type pair. And then finally, our last possible combination is another wild type pair. When we sum up all these odds, we can see that the overall probabilities, or 2 out of 4 times, we will have an FH negative, FH positive, meaning these babies will go on to develop HLRCC. The other 2 fertilization pairings will consist of 2 wild type alleles, meaning these babies will have perfectly working FH, so no risk of HLRCC. And just so things are clear, this plays out the exact same way if the father were to have HLRCC. There's still a 50-50 chance of a sperm getting the mutant allele or the wild type allele. So we can see how this plays through, just like before. And then when we look at the Punnett Square, we can see how these combine. We see that 2 out of 4 times we have a mutated allele mixing with the wild type, resulting in a positive genetic test for HLRCC, while the other half will have no mutated variant, and these offspring will have negative tests for HLRCC. And just as a point of clarification, in case anyone is confused by the positive-negative terminology, the genetic test says you're positive for a disorder, it means your DNA possesses a mutated allele. This nomenclature is going to be different from the FH positive, FH negative descriptors we see on the sides of the Punnett Square here. Those positive and negative signs are in reference to if you have a wild type allele or not. So this is why they are incongruent with the genetic results. They're referring to opposite reference points, either the presence of a mutation in the genetic test versus the presence or absence of a wild type allele in the Punnett Square example. Let's now take a look at what all this means for families with multiple children. It's very important to understand that each instance of inheritance is an independent event, meaning that the outcome of one event does not influence the outcome of another event. Just like flipping a coin doesn't change each time you flip the coin. It's always 50-50. In this case, the outcome of one child's genetic fate has no influence on subsequent children. And so the odds of inheriting the mutation for each individual child are always 50%. That being said, you can calculate the cumulative probability of the overall chance you expect to see in a group of children, but that probability in math gets somewhat complicated, so I'm going to save that for a separate video after this one. Now you might ask, what happens if both parents unfortunately have HLRCC? Here now we can see our dad now has one mutated FH allele. Running through our Punnett Square again, we can see all the potential pairings. Notice now something different is possible. 1 out of 4 children will have no functioning FH allele, and thus very little to no FH protein. This leads to a very disabling and deadly recessive genetic disorder called FH deficiency. This is an incredibly rare disease because of how unlikely it is for a man and woman with an FH variant to randomly meet and then have an FH deficient child. However, probability of this happening can significantly increase if a small insular community is founded by individuals who just happen to have had HLRCC. The gene pool of that small community will have a much higher prevalence of HLRCC, and eventually, if no outsiders join the community, there will be a bottleneck of genetic diversity, and genetically related people will get together and start having children. This highlights precisely why inbreeding is biologically dangerous from a genetic standpoint. The chances of combining two bad alleles is much greater when you reproduce with people closely genetically related. This is famously the case in the Short Creek Mormon community in Arizona-Utah area, which was founded just a few generations ago in 1930, where now an 8,000 person community is said to be 75 to 90% blood related to the HLRCC positive polygamous founders of the town. At a time when only 13 known cases of FH deficiency were known in the world, 20 more were discovered in the Short Creek community. I've linked a BBC article about this community in the description below. Looking back at our Punnett Square, there's a 50% chance of 2 HLRCC parents having a child with HLRCC. And then finally, there's a 1 in 4 chance, or 25% chance, of the child lucking out and not having any mutation in this case. Now, just to play through what happens after the sperm and egg come together. The fertilized egg now has all 46 chromosomes, and in this case, we can see it has 2 copies of chromosome 1, with one allele being the mutated and the other being the wild type. And then from this one initial starting cell, every other cell in your body will be copied from that original. And this means every cell in your body that contains a nucleus and DNA will contain both a good and bad allele for FH. So in our next video, we will discuss the consequences or the downstream effects of having cells that use both good and bad copy of the FH gene. Thanks again for watching, and feel free to ask any questions and leave any comments, and I'll see you soon.